I'd like to welcome you all to the 13th session of the International IVF Initiative. There are 1,061 people who have registered for this session. I am honored and excited to be co-hosting the International IVF Initiative with Tony Anderson. For videos of previous sessions, please go to ivfmeeting.com. You will also find information about upcoming sessions on that same page. Our continued goal is to support the IVF community during the COVID-19 pandemic and provide educational opportunities while bringing our community together. I personally have really enjoyed seeing so many reproductive biologists reaching out from across the world to participate in past sessions as well as today. I am Dara Berger and I will be moderating this session. We have an amazing program planned for the next 70 minutes. I want to thank the American Association of Bioanalysts for approving this program for continuing education credits. I encourage you all to ask questions using the question and answer session of the conference service. Please do not use the chat function for questions you want the speakers to answer. Any questions not asked to our speakers live will be answered and posted on our website, ivfmeeting.com, in the next few days. This pandemic situation has become one that none of us could have imagined. As a result, many conferences and educational activities have been canceled, including the annual CRB symposium that was going to be held in Orlando, Florida last week. Many scientists submitted abstracts, and we would like to thank each of them. The top 4 abstracts were chosen by the CRB Abstract Committee to be presented to you today. International IVF Initiative has a talented team who are working behind the scenes to post your questions to our speakers. I encourage you to ask your questions using the question and answer section of the conference service. Some questions will be addressed live and some will be typed into the question and answer section directly. All questions and answers will be posted online in the upcoming days. I will now turn the program over to Tony, who will introduce you to our speakers.
Thank you, Dara, and welcome everybody. It's an honor and a pleasure to be here today. And I would like to thank the International IVF Initiative for putting this together. As Dara said, our meeting would have occurred last week, and so it's a great opportunity to have these abstracts presented today, and they are approved for credits through the ABB for 1 hour. So I encourage you to get your certificate, submit those as you go. I would like to introduce our first speaker, Khalied Kaskar. The title of his presentation today is Growth Differentiation Factor 9, GDF9, Levels in Spent Culture Media and Pregnancy Outcome. Khalied has more than 25 years experience in embryology, starting at Tygerberg Hospital in South Africa and then Saudi Arabia, and currently Khalied serves as the lab manager at Baylor College of Medicine in Houston, Texas. He is also a faculty member of the Department of Obstetrics and Gynecology at Baylor. He is certified technical supervisor by the American Board of Bioanalysts in both embryology and andrology. If that doesn't keep Khalied busy enough, he serves as a reviewer for the journal Andrologia, and is also a Ph.D. candidate at the University of Western Cape in South Africa. It is my pleasure to introduce Khalied Kaskar.
Thank you, Tony. I'll share the screen. Okay. Good afternoon, ladies and gentlemen. I would like to thank the College of Reproductive Biology and the International IVF Initiative for this opportunity to share our research with you, especially during this difficult time with the pandemic. And I hope that everybody's doing well and staying safe. The title of my talk is Growth Differentiation Factor, GDF9, Levels in Spent Culture Media and Pregnancy Outcome. This is a collaborative study with Baylor College of Medicine, Texas Children's Hospital, the University of Western Cape in South Africa, Cleveland Clinic, and Ansch Labs. So what is GDF9? GDF9 is a growth factor that is secreted by the oocyte during folliculogenesis. It, along with others, They are members of the transforming growth factor beta superfamily, and they play key roles in promoting follicle growth, maturation, and oocyte competence. It's been shown to be expressed by both the oocyte and cumulus cells, and there are reports that show that GDF9 helps prevent apoptosis and follicular atresia. From this diagram, you can see this yellow line here that shows the activity of GDF9 from secondary follicles all the way up to ovulation, along with other growth factors like BMP15 that are crucial for follicular development and oocyte development. It's also been shown that the increased levels of GDF9 messenger RNA levels in cumulus cells have been correlated with oocyte maturation, fertilization, and also embryo quality. In addition to that, some reports have looked into adding exogenous GDF9 to in vitro maturation media, and have found that it actually improves oocyte maturation as well as embryo development. In contrast, the absence of GDF9 has been shown to cause sterility due to a block at the primary stage of follicular genesis. And any mutations or aberrant expression of GDF9 may be associated with polycystic ovarian syndrome, or PCOS, and may also cause premature ovarian failure. However, very few studies have shown if GDF9 continues to be expressed during embryo development. In a recent study by Liu et al. Looking at water buffalo, they have found that GDF9 is expressed during follicular genesis and also in embryogenesis. They further looked into the GDF9 of spent media of embryos and shown that GDF9 levels increase up until the 4-cell stage and then starts to decline at the 8-cell stage. They postulated that the high GDF9 levels could be remnants from follicular fluid that are still present in the oocyte, or it could be continued production of GDF9 from the granulosa cells, which later start to decline after day 3. Another possibility is that as the metabolic requirements of the embryo changes after day 3, as we all know it does, the secretion of GDF9 may also change. There has been no work published regarding the GDF9 secretion of the human oocyte after ovulation or fertilization. So the objective of this study was to measure GDF9 levels in spent culture media of human blastocysts and time-lapse morphokinetics and compare them to pregnancy outcome. We looked at 113 patients, all who have undergone ICSI with PGTA and frozen embryo transfer. We only looked at day 5 and day 6 blastocysts and excluded day 7s. We only looked at frozen transfers that had single euploid embryos transferred. We compared the GDF9 levels in the spent media and time-lapse morphokinetics in the different pregnancy outcomes, being those that did not get pregnant, as evidenced with a negative beta HCG 2 weeks post-transfer, biochemical pregnancies with a positive beta HCG, and clinical pregnancies were evidenced by a heartbeat 6 weeks post-transfer, and early miscarriage with no heartbeat after 6 weeks. Embryos were incubated in the EmbryoScope time-lapse incubator, and the time-lapse annotations we looked at were the time for the PNs to appear and to fade, and the various cell divisions thereafter from 2 cells to 8 cells, and also the time to morula, the start of blastulation, the time to blastocyst, and expanded blastocyst. With regards to spent media collection, we collected 22 microliters of media on the day of biopsy after the embryo was removed. The reason we chose 22 microliters is to avoid contamination from the oil in the dish. And then we also looked at the control media in the wells that did not have embryos, and they were also collected on the same day and served as our controls. The pipette tips were changed for each sample to prevent cross-contamination, and samples were placed in protein-free PCR tubes and stored at -80 degrees until the time of analysis. GDF9 levels were measured using a GDF9 ELISA kit. All measurements were performed at Ansch labs for both the control media and the spent media, and all assays were run blinded. So from the 113 FETs that we've had, 57 of them were with embryos that were biopsied on day 5, and 56 from embryos that were biopsied on day 6. We found a significant difference between the biochemical pregnancy rate and clinical pregnancy rates between embryos that were biopsied on day 5 versus day 6, from 80% to 62, and the same with clinical pregnancies. With regards to GDF9, we found a significant increase in a significantly higher level of GDF9 in day 5 spent media compared to day 6. And also interesting to note is in the control media that did not have any embryos, GDF9 was undetectable, going to show that the GDF9 values that we do see did originate from the embryo itself and not from the culture media. This graph illustrates the rapid decline in GDF9 from day 5 to day 6. We don't know when this decline occurs, whether it occurs in day 3, as was seen earlier in the water buffalo study, but we do see a very marked decrease. With regards to day 5 embryos, we looked at the time-lapse morphokinetics and pregnancy outcome. And we found no significant differences in any of the time-lapse time points that we looked at from the time that the PNs appeared till the time of expanded blastocyst between non-pregnant, miscarriage, clinical, or biochemical pregnancies. However, on day 6, we found significant differences in the non-pregnant and clinical pregnant groups from the time that PNs faded up to the 8 cells and start of blastulation and blastocyst. Interesting to note, however, the clinical pregnancies tend to develop a little bit slower than the ones that did not get pregnant, which is a little contrary to what the literature suggests. With regards to GDF9, as I mentioned before, the total GDF9 on day 5 was significantly higher than day 6. This trend was still evident when we broke them down into the different pregnancy groups, into the biochemical and clinical pregnancy groups. That elevation was still seen in day 5, and they were significantly different. However, when looking at day 5 per se, there was no significant difference in the GDF9 between the various pregnancy groups, the negative, biochemical, clinical, and early miscarriage. The same happened for day 6 as well. No significant differences were seen between the pregnant groups. In conclusion, we found that the day 5 blastocysts showed much higher biochemical pregnancy rate, clinical pregnancy rate, and GDF9 values in spent media as compared to day 6 blastocysts. And also the day 5 blastocysts showed no significant difference in any of the morphokinetic parameters between the pregnancy groups. In day 6 blastocysts, the non-pregnant group showed significant differences in time-lapse morphokinetics compared to the clinical pregnant group in all time points up to the 8-cell stage and start of blastulation. Interestingly, the clinical pregnancy groups showed slower time-lapse embryo development than non-pregnant groups. Why that is, we do not know. With regards to GDF9, which was the initial objective of this study, we found that the assay we used could detect GDF9 in as little as 22 microliters of spent media. The GDF9 was undetectable in the control media, showing that GDF9 originated from the embryo itself. However, the GDF9 was not able to show any significant differences between the pregnant and non-pregnant groups of day 5 or day 6 embryos. The lower daily GDF9 values that we found in day 6 could be a possible explanation for its slower embryo development. And that would require some more research to look into that. Also, the decline in GDF9 on day 6 could also indicate that GDF9 production declines after a certain point. What that point is, we don't know yet, and I think future studies will be looking into that. So due to the small sample size of this study, more research needs to be done to understand the influence of GDF9 on embryogenesis, and since very little is known about the embryogenic activity of GDF9. With that, I thank you for your time and your attention.
Excellent presentation, Khalied. Thank you so much. We have some questions coming in, and while the next presenter comes in, we'll ask a question here. We have a question. Since this was a quantitative study, how were you able to correct for different length of culture between day 5 and day 6 embryos when analyzing media?
Well, on day 5, whenever we, the embryoscope itself has got 4 rinse wells, which we don't typically use. So whenever we take out an embryo on day 5, we take out a corresponding control media on day 5 as well. And we do the same on day 6. So we know that the control media that we have for that corresponding embryo or cohort of embryos that we do on day 5 will have a corresponding control value as well.
Excellent. Yeah, it'd be great to use this for testing and also with graduated embryo scoring, combine it all together. I think it's a very good presentation. Thank you so much.
Thank you.
We'll come back and ask some more questions. I would like to remind everybody, put your questions in the Q&A. We see some people raising their hands, but go ahead and just type your questions in there. Our excellent group of panelists are managing those questions, and we'll get to most of them at the end of the session today. Our next presenter is Dr. Renee Chost. The title of her presentation today is Isolation, Purification, and Initial RNA Sequence Analysis of Seminal Fluid Exosomes between Pregnant and Non-Pregnant Intrauterine Insemination Pregnancies. Dr. Chost is a clinical assistant professor of biochemistry in the Department of Biomedical Sciences at the University of South Carolina School of Medicine in Greenville. She is also the director of medical student research at the university. She earned her Ph.D. in biological chemistry from the University of Texas Southwestern Medical Center at Dallas, followed by a postdoctoral fellowship at M.D. Anderson Cancer Center in Houston, Texas. Dr. Chost is a trained biochemist and molecular biologist who recently changed her research area to reproductive biology to study infertility at the molecular level. She uses a biochemical and molecular techniques to identify potential indicators of implantation outcomes in both blastocele and fluid and seminal fluid. And with that said, I present to you Dr. Renee Chost.
Thank you so much thank you so much for the opportunity to present today, and thank you all for being here this afternoon. As Tony said, this project is an RNA-seq project analysis on seminal fluid exosomes between pregnant and non-pregnant IUI insemination pregnancies. And I'm a professor at the University of South Carolina School of Medicine in Greenville, South Carolina, and that's where this work was performed. This project is a collaboration with BIOS Fertility Institute as well as Clemson University. All authors have nothing to disclose. The objective of this project was to compare the RNA content in exosomes purified from seminal fluid associated with IUI pregnancies with the known pregnancy outcome. The design of the study was a retrospective analysis of exosomes from RNA from seminal fluid from patients undergoing IUI with known pregnancy outcomes. What was interesting about this study and made it different is that we utilized a new method for isolating these exosomes, and that was hydrophobic interaction chromatography, which I'll go into in just a minute. So what is known about the RNA content in seminal fluid exosomes? Well, many people think that exosomes are signaling vesicles that just contain microRNAs. However, This study that came out in 2014 showed that exosomes carry a variety of RNAs, including microRNAs, ribosomal RNAs, tRNAs, and other RNAs. And I will note that the exosomes purified for this study were done using ultracentrifugation, which is the typical way that exosomes are purified from different fluids. In 2016, another study came out in fertility and sterility that characterized microRNAs from seminal fluid exosomes from men with poor motile sperm versus men who had normal motile sperm. And this study identified microRNAs that were expressed in spermatozoa that were associated with signaling pathways that had roles in spermatogenesis. And another study came out in 2018 that was in human reproduction that looked at microRNAs and exosomes from men with normal sperm or men with azoospermia. They identified microRNAs that were downregulated in the azoospermia samples and found that looking at microRNA exosomes, this may be a non-invasive biomarker to diagnose the cause of men who aren't producing any sperm. And I will mention both of these studies were done by purifying exosomes, again, with ultracentrifugation procedures. So in our lab, we were looking to characterize the RNA content in seminal fluid and in blastocele fluid, and we were about to start doing some traditional exosome purification methods. However, we learned that Clemson University, a neighbor to our university, had developed a novel way to purify exosomes from cervical mucus samples that involved a hydrophobic interaction chromatography. So we were able to collaborate with them using some IUI seminal fluid samples from VIOS. We worked with Clemson to purify these exosomes and then performed RNA-seq on these exosomes. So briefly, the group at Clemson is led by chemist Dr. Ken Marcus in collaboration with Dr. Terry Bruce, and they developed an HPLC-based method to purify exosomes from various fluids. So the way this process works is you inject your fluid into the HPLC in a high salt buffer. The high salt buffer is going to allow these hydrophobic regions to be exposed, and they will bind to a polyester column that Dr. Marcus has developed. And then you decrease the salt concentration in the buffer, and that allows the proteins and exosomes to then fall off of the column. So the graph on the bottom right here shows the initial elution from this polyester column is going to be peptides and proteins. And then when you lower the salt concentration, the next peak is going to be exosomes coming off. And what's wonderful about this procedure is the time frame here. So these exosomes are purified in less than 10 minutes. So this is a huge deal for the type of project we're doing, where we're going to be purifying exosomes from many samples. The other benefit of this chromatography as opposed to ultracentrifugation is we're not worried about damaging the membrane of these exosomes. So as I mentioned, the fluid samples that we used were collected at VIOS. These samples were matched with total motile counts per insemination, and the IUI outcomes are recorded. And we grouped the samples into two groups. We had 6 seminal fluid samples associated with positive outcomes and 6 seminal fluid samples associated with negative pregnancy outcomes. Before we sent the exosomes off for RNA-seq analysis, we confirmed the presence of these exosomes using transmission electron microscopy. And what was interesting is when we looked at the exosomes, the positive IUI outcome versus those from a negative IUI outcome, we saw that those associated with the positive IUI outcome had a large number of these very small exosomes in the 30 to 50 nanometer size, versus the negative IUI outcome exosomes didn't have a large number of these smaller sized exosomes. And we're really not sure what this means at the physiological level. So briefly, we then took these purified exosomes that were purified using this hydrophobic interaction chromatography, and we worked with SBI System Bioscience. And the first thing they did was purify the RNA from these exosomes and then constructed libraries from the RNA and then sequenced these libraries and aligned the libraries with the human genome. So we were able to look at RNA copy number based on the different types of RNAs. So microRNAs, link RNAs, mRNAs, et cetera. And then finally, we were able to compare these sequencing results from the positive IUI pregnancies to the negative IUI pregnancies. And this is a brief overview of what we found. The top shows the 6 positive pregnant samples, and the 6 negative samples are on the bottom. And notice in this green color, we had tRNAs present in quite a large amount in all of our samples. And for microRNAs, the light blue color, we saw them expressed again in all of our samples. So just to look a little closer at the data, we found that the abundance of microRNAs was actually greater in the exosomes from non-pregnant samples versus pregnant samples, and this was statistically significant. We also looked at link RNAs, which not everyone is much familiar with. So link RNAs are these long energetic non-coding RNAs. So they also have a regulatory function for transcriptional regulation and chromatin regulation. So these link RNAs were also in a higher abundance in our non-pregnant samples versus our pregnant samples. And finally, we saw that coding mRNAs that actually give rise to gene products were actually at a higher level in our pregnant samples versus our non-pregnant samples. So overall, we showed that microRNAs were expressed in greater abundance, as well as link RNAs in the non-pregnant samples versus our pregnant samples, while coding mRNAs, we saw greater expression in pregnant samples. So why would we see greater amounts of coding RNAs in these pregnant sample of exosomes? Well, it's possible that these mRNAs need to be present in the seminal fluid to aid in fertilization later on, because these coding mRNAs give rise to gene products. So overall, we were able to purify seminal fluid exosomes using this new technology of hydrophobic interaction chromatography, which hadn't been reported to date, that we found in the literature, and that the RNA content differs from these exosomes versus pregnant versus non-pregnant samples. So our further studies that we're going to be doing is looking further at the actual identity of these microRNAs, link RNAs, and mRNAs that are associated with these various pregnancy outcomes. And we're doing this in collaboration with a bioinformatics researcher, Dr. Alyssa Clay Gilmore, because we want to identify a unique set of RNAs in each group. And then what we will be doing is verifying the expression of these RNAs in a whole other set of seminal fluid samples using real-time quantitative PCR. We're most interested in the mRNAs. There hasn't been as much research in this area from seminal fluid exosomes. So we want to select a group of mRNAs that are unique to the pregnant samples versus non-pregnant samples, and then verify those using another technology. We're also very interested in looking at the proteins present in the membrane of these exosomes. These exosomes were purified in a way that maintained membrane integrity. So we're very interested in understanding which proteins are present in those membranes. We also want to follow up on this study where we saw on the transmission electron microscopy that exosome size and number differed between our samples. So we want to further look into what's going on here. We're pretty sure that the HPLC technology we use to purify the exosomes won't be able to separate them by size. So we're looking into another method of separation. And finally, the goal of this project really isn't to improve IUI rates. The goal of this project is the main goal of our research, which is to identify molecular signatures in seminal fluid as well as blastocell fluid. So by molecular signatures, I mean RNAs and proteins and genes that are associated with positive or negative pregnancy outcomes to eventually develop screening processes to improve IVF success rate. So with that, I'd like to thank my collaborator and project principal investigator, COI, Dr. William Raudebush, as well as Dr. Sean Zimmerman at VIOS, Dr. Ken Marcus and Terry Bruce at Clemson and their graduate student, Sissy, and then our 2 medical students, Emily and Weber. And thank you very much for this opportunity.
Thank you so much, Renee. That was a great presentation. So our first question for you is, did the total content of RNAs correlate to the increased presence of exosomes you identified on TEM?
we don't know that. We haven't gone back and looked at the TEMA for all the different samples we analyzed, but it's definitely something we need to go back and do for sure. It's a great question.
Okay, great. We'll circle back to you again with some more questions at the end. Tony, would you like to introduce our next speaker?
It would be my pleasure, Dara. Our next speaker is Dr. Brent Hanson. The title of his presentation today is The Relationship Between Age and Rates of Abnormal Fertilization Following Intracytoplasmic Sperm Injection. Dr. Hanson is currently a reproductive endocrinology and infertility fellow at Reproductive Medicine Associates in New Jersey. He received his Bachelor of Science degree in physiology from the University of Arizona before continuing his education at West Virginia University School of Medicine. Following medical school, he completed his obstetrics and gynecology residency training at the University of Utah in Salt Lake City. And during his infertility fellowship, Dr. Hanson had the opportunity to work extensively in the embryology lab. His research interests primarily include male factor infertility, cryopreservation and vitrification techniques, and changes associated with reproductive aging. It is my pleasure to introduce Dr. Brent Hanson.
All right. Thank you very much for that introduction. I'm really glad to be here today to present our research. I speak on behalf of our co-authors just to say that our lab does a lot of great work every day, and we're glad that we can highlight some of that today. So like you mentioned, the presentation that I have today is the relationship between age and rates of abnormal fertilization following ICSI, which really links the clinical problem of older patients or aging patients with the lab problem of poor fertilization. So I have nothing to disclose. And when we look at this as a topic, obviously we know that normal fertilization after ICSI is confirmed by the presence of 2 pronuclei, with each pronucleus containing genetic information from a single parent. And in our lab and most labs, that check is done about 16 to 18 hours after ICSI is performed. We also know that zygotes that exhibit a 0 PN or failure to fertilize, 1 PN or 3 PN or greater state indicate some form of fertilization failure or abnormal fertilization. And with that, high rates of failed or abnormal fertilization may indicate problems within an embryo cohort related to the quality of the gametes. So we wanted to look at this population specifically and see if age factors into these fertilization outcomes. With normal fertilization, the ICSI successful fertilization rates are usually above 70%, with most labs and clinics reporting somewhere between 80 and 90%. And while failed fertilization, where nothing fertilizes, is relatively rare, comprising about 3% of ICSI cycles, it is common for patients to have a few abnormal or failures within the cohort. When a 0 PN is noted, typically that's an egg that remains at the M2 phase, and that would be a result of a failure of the oocyte to activate or possibly a failure to actually inject sperm. A 1 PN result, which happens in about 1% of ICSI cases, results from DNA that's missing from one of the gametes, or a case where the oocyte is activated, but the sperm is defective. And as a result of this, this is not successful, but can exhibit 1 or 2 polar bodies, just depending on what exactly went wrong, or if it's a timing issue. 3 PN embryos take up about 1% of all ICSI cases as well, and can result from polyspermic fertilization, which we shouldn't see in ICSI, but that could be a potential in conventional insemination, or potentially the retention of a polar body. So with all that being said, the question here is, does age impact the rates of abnormal fertilization? Studies have looked at this, and we took this both from the male side as well as the female side. So starting with paternal age or male factors, we know that advanced paternal age is associated with increased DNA fragmentation and worsened semen analysis parameters. And the impacting of increasing male age on ICSI outcomes is relatively conflicting. So in 2009, there was a study that came out looking at rates of fertilization and found that they were lower with conventional insemination if men were older than 50 years of age. In 2007, a similar study looked at the same question with ICSI and found that fertilization rates were lower if men were older. Spandorfer in 1998 found that there were higher rates of 3PN fertilization in ICSI if men were older, over the age of 50. But the problem with all of this literature is that while we can identify studies that support this claim that maybe fertilization is abnormal in older men, many studies have found no difference in embryology outcomes or fertilization rates in an older population. When we take this issue looking at the female perspective or the female side, clearly advancing female age has negative effects on fertility, and there's a natural decline in female fertility throughout the lifespan. And this relates to both the diminished number of oocytes as well as the decreased quality of oocytes. And this question about whether fertilization occurs like it should in older patients or older women really deals with the factor of oocyte quality. So when we look at the relationship between female age and oocyte quality, we can assess that relationship in many different ways, and it has been looked at in many ways. But the data, once again, are conflicting about what is the true impact on each of these factors. And part of that has to do with whether you're looking at embryology outcomes or pregnancy outcomes. And we can really take female age and see if it impacts all of these different areas, whether maternal age plays a role in euploid stage or arrest of embryos, decreased live births. So there are a lot of ways to look at this. But for this study specifically, we wanted to focus on fertilization. It has been reported previously that advancing maternal age is associated with increased rates of 3PN zygotes in both conventional IVF and ICSI, where rates of normal fertilization, at least the percentage of 2PNs present, were not affected by age. So for this study, what we looked at was evaluating the relationship between age for both male and female partners and how those embryos fertilized by looking at the rates of each of these factors. And the question really that we were trying to answer was, do older patients exhibit higher rates of abnormal fertilization as a result of diminished gamete quality? So this was a retrospective study looking through our electronic medical records. We looked at all of our cycles between 2012 and 2019 at our practice, and all cycles included were done, fertilization was done via ICSI, and we used only fresh ejaculated sperm. We evaluated the rates of each outcome measure, the 0PN, 1PN, 2PN, or greater than 3PN. And for statistics, we used linear regression models to control for the age of the opposite partner. So looking at these cycles, we ended up evaluating 8,300 cycles. For the male partners, the average age was 37.7. The female partners from this group, average age was slightly less, 35.4. Overall, you can see that our fertilization rate for successful fertilization or normal fertilization was 84.4%. And then about 16% fell into the abnormal fertilization category, with 0PN being the most common abnormal outcome. So does age matter? When we look at this from the male side, we use the male age group of less than 30 as the reference group and compared all age groups to that group. So for the male age in the 30 to 34.9 category, there was no statistically significant effect seen with aging in terms of fertilization outcomes. Similarly, for the 35 to 39 group, there was no statistically significant effect on fertilization outcomes as men aged. And the over 40 group, kind of the same story. So as we compare these graphically, we can see that looking at the 0PN, 1PN, 2PN, and 3PN rates, they were relatively similar across all age groups. And the bottom line here was that male partner age did not seem to affect rates of abnormal fertilization. Looking at this from the female side, we asked the same question, again, using an age less than 30 as the reference group. And we found that for the 30 to 34-year-olds, we did not have a statistically significant impact. For the 35 to 39-year-olds, this was where our data became a little bit interesting because there were no differences in rates of 0PN, 1PN, or 2PN embryos, but we did see an increased rate of 3PN embryos for this 35 to 39-year-old female age group when compared to the less than 30 group, which was our reference. For the greater than 40 group, however, there was no statistically significant difference. Looking at the same style of graph here for female age, you can see again that our statistically significant differences were not seen across most of these outcome measures. And even for the 3PN outcome, which we did have a significant difference between the less than 30 age group and the 35 to 39, it's only 0.6% of embryos to 0.9% of embryos. So really the absolute values there are relatively similar. So the bottom line, compared to women less than 30 years old, women between the ages of 35 and 39.9 did have statistically significant higher rates of 3PN zygotes, and that finding was not observed in women less than 40. So we may be able to say from this that rates of abnormal fertilization were unaffected by increasing age of either partner, and a woman's age may impact early embryo morphology because we did see that higher rate of 3PN embryos, but the absolute value, like I mentioned, was relatively small, only 0.6 versus 0.9%, and the trend was not observed in women over 40. So if our thought was with increasing age, we should see more of this abnormal fertilization, that wasn't the case. Some of the limitations here, this was a retrospective study, and our data analysis assumes that the youngest group, the less than 30 group, is the ideal reference population, which ignores the fact that there may be potentially compromised performance at the extremes of age. So we don't necessarily know for certain if the less than 30 age group is the ideal reference point. And different age cutoffs may lead to different findings, and overall, a relatively small fraction of embryos fall into this 3PN category, so it's challenging to make strong conclusions. Going forward, we can further investigate the relationship between age and abnormal fertilization rates, clarify how age impacts other aspects of embryo development, and really look at why this is occurring. If oocytes have a compromised ability to extrude a second polar body, that may be one issue, and then a compromised polyspermic block could be an issue with conventional insemination, but shouldn't be an issue with ICSI. And really just seeing if these findings hold true with larger patient populations or other situations like conventional insemination, surgically extracted sperm, or previously cryopreserved gametes. Thank you to our entire team and our lab. You know, it's been a great experience working with everyone in our lab, and I know that they work hard every day. So I really appreciate their input and help with this.
Thank you, Dr. Hanson. Very good presentation. Stimulating the neurons there for sure. If I were to think about it, we heard a lot in some of these presentations about DNA fragmentation, processing of sperm. You know, as men age, you know, the theory would be that they would contribute some of this, but your data was very, very well presented. You mentioned at the end about conventional InStem. What are your thoughts on that? I know that this was all ICSI, but you know, would the results be? Would you expect any different results if you use conventional insemination?
I really don't. You know, I don't know for certain because we didn't look at that, but I don't think the results would be different. I do think that maybe some of the mechanisms for why we're ending up with a 3PN embryo may be different. You know, but I know every lab is also different in their rates of ICSI versus conventional InSem. For us, because we do so much ICSI, I think the more important question was to look at ICSI, but it's definitely something that could be studied in that population.
Yeah, we do all ICSI in our lab here in San Antonio, but there's a good argument for not to do all ICSI too. Like some eggs just wouldn't fertilize, rather, you know, you're getting your best embryos that way. But I still like to do ICSI. Just my personal preference.
Yeah.
Our next speaker is going to be Kathryn Wozniak. And while she's putting her screen up, Kathryn's title of Kathryn's talk today is Clinical Benefits of Culturing Monopronucleated Zygotes Derived from Intracytoplasmic Injection Cycles. Kathryn Wozniak is an embryologist at Ovation Fertility in Newport Beach, California. In 2016, she earned a Bachelor of Science at Monash University in Melbourne, Australia, With a keen interest in embryology, she went on to complete an honors research degree in male infertility laboratory, and in 2018, she was awarded a Master of Clinical Embryology. Throughout her time at Monash University, Kathryn received numerous scholarships and academic rewards. And to that said, I'm introducing Kathryn Wozniak.
Thank you, Tony, and good afternoon, everyone. We would like to thank the organisers of the International IVF Initiative and today's moderators of the AAB conference webinar. We are honoured that the CRB Abstract Committee selected our study for presentation. So on behalf of my lab director, Mitchell Sheely, and I, today I will be talking about our experience looking at the clinical benefits of culturing single pronuclear zygotes derived from ICSI cycles. In applying today's assisted reproductive technologies, the authors have no conflicts of interest to disclose. So historically, all ART laboratories see single pronuclear zygotes occurring about 5% of the time. The established timing of pronuclear appearance has strongly influenced how we think about the mechanisms proposed to result in a 1PN embryo. So if we think about a traditional IVF insemination model, we don't know exactly when sperm penetration has occurred. Consequently, it has been theorized that the presence of 1PN may be due to an asynchronous appearance or disappearance event. Therefore, in an IVF system, it is uncertain whether 1PN zygotes are normal or in fact abnormal. If we compare this to an ICSI fertilization system, sperm penetration is a timed event. Therefore, it has been assumed that 1PN formations result from an abnormal fertilization event. So our lab has been performing ICSI only for over 15 years. And based on this model, we have typically considered our 1PNs to be abnormal and discarded them on day one of development. So recent publications have, however, confirmed that 1PN zygotes do have the potential to be chromosomally normal. In these studies, although 1PN blastocyst rates are shown to be as low as 15%, euploidy rates have been reported to be over 50% in these embryos. So much of the clinical work looking at 1PNs has focused on those arising from IVF. However, healthy live births have been reported after the transfer of both normal IVF and ICSI-derived 1PNs. So this suggests that 1PN blastocysts could be of clinical value if we can select against the abnormally fertilized ones that are associated with poor pregnancy outcomes. And based on this data and the fact that we perform PGTA on over 85% of our patients, we thought this was a good opportunity to validate our experience with 1PN embryos. So in this study, we aim to evaluate the developmental competence and aneuploidy status of blastocysts derived from 1PN zygotes following ICSI and PGTA. So just quickly, before I continue, I just want to ask the audience a few questions. I'm interested to firstly know whether your lab discards your 1PN zygotes derived from ICSI on day one of development. And secondly, if you were to culture 1PN zygotes to the blastocyst stage and they came back with a euploid result, would you consider that embryo for transfer? So this diagram shows a basic outline of our study design. ICSI was typically performed 2 to 4 hours post-retrieval, and pronuclear assessment was carried out 16 to 18 hours later. So patients with 1PN zygotes were included in our study. Their zygotes were subdivided by number of PN and cultured to day 3 of development. And so at this point, all embryos underwent laser-assisted hatching. Blastocyst formation was assessed on days 5 through 7, and all fair to good quality blastocysts were biopsied and or vitrified. The trophectoderm cells were sent to our genetics team and they performed PGTA using next generation sequencing technology. Each embryo was then classified as being euploid, mosaic or aneuploid. So I guess we were really interested in 3 key indications of embryo quality. We assessed each embryo's ability to form a blastocyst, its morphological quality, and also genetic status post-biopsy. So we obtained a total of 75 1PN zygotes from 54 fresh ICSI cycles, representing 27% of our total fresh ICSI cycles. From this table, you can see that compared to the 2PN cohort, our 1PN embryos not only had a lower rate of blastocyst development, but also a reduced ability to produce a good quality embryo capable of biopsy or vitrification. If we go on to look at the day of embryo biopsy, we can also see that our 1PN embryos were less likely to be of suitable quality on days 5 and 6 of development, with higher delayed development on day 7. So as our PGTA reports came back, we were continually surprised at the number of 1PN embryos showing a euploid result. We also noticed that many of the 1PN genetic reports were coming back as 46XX or normal female karyotypes. So our 1PN euploid rate did turn out to be higher than that of the 2PNs, with a 3-to-1 skewing of normal female. So this deviates from the typical 50% ratio of male to females that you can see in our 2PN cohort. So taking this into consideration, I guess we thought there must be a logical explanation for this. So when we use PGTA technology as a screening tool, it is important to remember what this technology tells us. So next generation sequencing uses copy number ratios to detect common abnormalities of individual chromosomes. So it cannot detect genome-wide numerical abnormalities such as haploidy or uniparental disomy. And this, I guess, must be taken into careful consideration, especially in a 1PN cohort where the risk of these abnormalities is high. So the appearance of an XY sex chromosome combination can be used to rule out these abnormalities in our male 1PNs. However, for the female 1PNs, we would need to do further testing to determine a biparental genome. Other groups have shown that about 15% of analysed 46XX 1PN embryos are indeed of parthenogenetic origin. So I guess with this in mind, we could speculate that the 3-to-1 ratio of euploid female to male 1PNs is in fact due to the presence of true monopronuclear females, which have resulted from an abnormal fertilization event. So in comparison with the current literature, our 1PN blastocyst formation rates were higher. However, this was also true for our 2PNs. We saw a consistent 20% increase across groups, suggesting that this may be a result of differences between culture systems. Our rate of good quality 1PN blastocysts was similar to what other groups have found. However, our euploidy rate was higher. And we do believe that our small sample size is a contributing factor in this difference. So in light of our results, we are continuing to let 1PNs prove their developmental competence by growing them to the blastocyst stage. Although we have shown that 1PN embryos have a reduced developmental potential, for some patients, these embryos may provide them with their only clinical suitable embryo for transfer, and therefore would give them a chance at obtaining a pregnancy from that cycle. So we do hope that in the future, healthy live births will derive from some of our 1PN embryos. In the near future, we hope to perform paternity testing on all of the euploid female 1PN biopsy samples to further delineate the normality of our 46XX blastocysts. So I guess obtaining this information will expand our knowledge on the clinical value of 1PNs And it would also allow us to inform our patients about the likelihood of a 1PN embryo producing a healthy live birth. So thank you all for listening today. I hope you enjoyed my presentation. And if anyone does have any comments, ideas, or 1PN data of their own, I would absolutely love to hear from you. So I have included my email down the bottom of this slide.
Thank you so much for your talk. Do we have the results of those survey questions up yet?
Okay,
Kathryn,
so
would you like?
yeah, so for the first question, does your laboratory discard ICSI-derived 1PN zygotes on day 1? 46% said yes, and 49% said no, with 5% not applicable. So I guess that is a really interesting result. I thought it would be more skewed towards the yes. So yeah, that's interesting.
Okay.
And
oh, go on.
The second question, so if you keep 1PN eggs in culture and it becomes a good quality blastocyst and you biopsy it and it is euploid, would you consider it for transfer? So 65% said yes, 13 no, and 22% was not applicable.
Okay, thank you for reviewing those. And for our question, were the number of polar bodies considered for your analysis?
So that's a great question. We actually did not record polar body excretion on day 1. So I guess moving forward, that would be a really great thing to record with our 1PN embryos so that we can compare that when we get our full genetic results, especially for the 46XX females.
Okay, great. So it looks like we have about 10 or 15 minutes left, and Tony and I are just going to go back and forth between our speakers and try to address some of the questions that our audience members have been asking.
Yes, my first question I'm going to shoot out to Khalied, and the question is, what do you think could be a possible reason for an increased time to clinical pregnancy for day 6 blastocyst? Khalied? Are you there?
Yeah, I'm sorry. I had it on mute. No, I think that's a great question. The short answer to that is we don't know. But I think that sometimes embryos that are developing a little bit too fast might also not be a good thing. I think research has shown usually embryos that are the fastest to blast are the ones that tend to have better pregnancy results. And then also that for every delay in getting to blastocyst stage reduces the pregnancy rate in some studies. So I think that sometimes we see embryos that are developing a little bit too fast, especially like on day 3, if you have more 10 cells embryos or 12 cell embryos on day 3, those are not usually indicative of a good embryo because they get moving a little bit faster than they should be. So that could be one explanation that I can put out there.
Yeah, great point. I always say anything greater than 10 cells is an indicator something's really not going right. If you have a lot of them, and likewise, if they're really slow too, you'll probably see some variability there as well.
Right.
I'm going to jump in and ask the next question to Renee. One of our viewers wants to know if you correlated RNA with sperm parameters.
Yeah, so that's a great question. So for this preliminary study, we didn't. We only had the motility. We didn't have morphology. We didn't have chromatin content, but we plan to do that as soon as we can get more samples from the lab. But yes, excellent question.
I'm going to send this question to Dr. Hanson. Do you consider did you consider conducting a similar study in IVF patients to correlate RNA sequence with sperm function and the ability to fertilize and subsequent embryo development?
That's interesting. We have not looked at sperm specifically for that question. But yeah, that's something that could be done. And, you know, there's a lot of great questions related to this issue of aging and how that affects fertilization and looking at the same outcomes with different inputs.
Thank you.
I guess we're kind of just going in order. We didn't plan it this way. But Kathryn, does any chance of a 1PN embryo have missing to check another PN, whether the 1PN embryo was originally a 2PN?
There is a chance that that could have happened and maybe one of the PNs disappeared or appeared before the other one. I guess it's less likely in an ICSI fertilization system because we know exactly when sperm penetration occurred, so we can check for fertilization at a time point when we know the pronuclei appear. So in saying that though, it could still happen. So yes, there is a chance that some of our 1PNs that has happened, and they are in fact 2PNs.
Excellent answer. I'm actually going to go back to Brent, Dr. Hanson. Have you done PGTA analysis on any of the 3PN embryos? Even though it was a small number, have you ever done PGTA on any of those?
Not in a formalized study. I mean, I don't have those numbers in front of you, what the percentages would be, but I know we have looked at a few individualized 3PN embryos, but not for clinical use or not in a formalized study.
Yeah, it's an interesting question because we've done some, you know, if you ask the PGT companies what percent are triploid, there's actually a fair number of triploid, even with all of our extra checks and things. So yeah, that would be an interesting evaluation.
Yeah, and I think, you know, obviously this relates to what Kathryn's doing too, but, you know, this identification of what is actually successful fertilization. We use these 0PN, 1PN, 2PN because it's what we have. But, you know, when we double check that with the genetics, it doesn't always line up. So there may be a timing issue or something else going on that makes this harder.
Yeah, of course. And 0PNs that divide have made lots of pregnancies over time.
So I have to let everybody and Renee know, my cell phone has been blowing up about your cat. Everybody seems very interested in your cat as well as your talk. So the next question for you is, what is the correlation of exosome RNAs and sperm quality?
So again, we did not assess that. We got really lucky with this study because we only had motility data to look at and actually saw differences. So our plan is to take what we have now and get the identity of these mRNAs and then go back, and then we can use real-time quantitative PCR to look at a huge number of samples, and then we can compare all sorts of things like chromatin content, morphology, and have a much better idea of what's going on in these samples.
Great. This question goes to Khalied. Would you ever consider adding exogenous GDF9 to the embryo media to promote embryo development?
I would. I'd probably start doing that in a mouse model first and see if we have anything there. If we show any improvement, we might try to get approval to do that in a human study.
Yeah. Interesting.
So I have a general question. This is from Rich Rollins. In some vertebrate species, haploid female embryos lead to viable pregnancies, but not necessarily live births. What is the case for parthenogenic haploid female human embryos, not viable compared to conventional XX from male and female gametes? Is there anybody out there who wants to take a crack at these questions?
So was the first question they wanted to know was if it's a haploid female, what will happen if we transfer it?
Well, he's saying that they lead to pregnancies, but not necessarily live births. So what is the case for parthenogenic haploid embryos?
Yeah, that's a tough one, I guess. I think they can implant, but they would miscarry if they're haploid. I know that if it's a male-only genome, so the oocyte hasn't contributed to the genome, that does cause molar pregnancies. So I guess, yeah, it's not ideal to transfer a haploid embryo. Does anyone else want to add on to that?
Yeah, I agree with you. I mean, I think, you know, unfortunately, it's not going to be successful. Kind of when that development stops may vary a little bit, but ultimately either an arrest during embryogenesis or a miscarriage or a negative pregnancy test.
Mm-hmm.
That's actually a really good point, because the thing that always drives me crazy is that even with all the checks and all the PGTA testing, we still have pretty high miscarriage rates after we get an embryo implanted. And it's one of the things I'm really loving these presentations for. It helps you kind of look into those ideas as to why that could be occurring. Looking for that magic pill. All right. I believe this question goes out to Renee. Dr. Burma says, thank you for a fantastic presentation. It would be interesting to see if any relationship between DNA fragmentation index and other microscopic RNA signatures for pregnant and non-pregnant outcomes.
Yes, those similar questions to before. Yeah, we don't have that data. We only have the motility data. And yes, great suggestion. We plan to do that for sure once we get more samples and look at directed specific genes once we have the CASA data, the chromatin integrity, and all that data. 100% agree.
Excellent. Thank you so much.
And I think we have time for one more question, and then we'll wrap up. Tony, do you want to pick your favorite that's left?
My favorite.
Your favorite question that's left.
My favorite question. Well, where was that? Actually, I saw a question here that I really was interested. I think it was, look on here. Having a hard time finding it. So this is for Khalied. And is there a side effect on the embryos if the GDF9 level is high? Similar to the exogenous addition, could there possibly be too much GDF9 in the media?
In animal studies, they actually have shown that too high of a GDF9 can have a negative effect on oocyte quality and subsequent embryo development if the GDF9 levels are too high. So yes, so if we were to try and do that as an exogenous addition to the media, yeah, we're going to have to sort of do a dose-dependent study to see what exactly is the ideal concentration that we would need to add. Because we wouldn't want to overshoot that.
Yeah, no, finding that perfect balance, right?
Right.
So, yeah, as Dara said, we're running out of time here. I would like to throw out a public service announcement. We have a great presentation, kind of a different idea this Friday, and Giles Palmer is presenting a session on Friday on mental health in the embryology lab. And there's a survey on the site at ivfmeeting.com. Check the upcoming meetings for this Friday. 1, register for the meeting. And 2, there is a survey. Just basically talks to you about handling the stress in the lab. Even though the COVID stress is part of what we're dealing with, we deal with stress every day. And this is going to be a great presentation on how we handle those stresses in the lab. So please go to the site and register for that. I had the pleasure of planning the CRB meeting this year that got canceled. And so I thank the IVF, International IVF Initiative and CRB for allowing us to do this. And Dara, the outgoing president, will plan next year's meeting in Austin. And I thank the panelists and everybody here today.